Everyone. I'm Hillary Andrews, the Director of Regulatory and Research Partnerships at Friends of Cancer Research. Thank you so much for joining us today for our Friends Annual Meeting 2024 recap, sharing next steps and opportunities with advocates. We're very excited to have a panel of experts, including representation from the FDA, pharmaceutical companies, academia, and our Friends Advisory Advocate Group. Our goal of this event is to review key takeaways from our annual meeting and provide you with an opportunity to ask questions of the experts. Please use the Q&A feature throughout the webinar to ask questions. Now I'll pass it over to our moderator, Nevine Zariffa from the NMD Group. Nevine is a consultant who serves as a working group member on both the ctDNA working group and the Intermediate Overall Survival working group.
Hillary, thank you. And I'll add a warm welcome to Hillary's to our panelists and also to all of you who are joining us here today. Let me quickly introduce the panelists, and then we'll do a bit of a recap on the topics that were discussed at the annual meeting, and then we'll turn it over to you for your Q&A, so don't hesitate to start populating with questions as they come up. So our first panelist is Alex Wyatt, who is an associate professor at the University of British Columbia in Canada. Joining him from the Circulating Tumor DNA Working Group is Carol Vallett, who was one of our patient advisory advocates. For the pragmatic trials, we have two panelists, Ashita Batavia, who is Head of Hematology Oncology Data Sciences at J&J's Innovative Medicines Company, as well as Ava May, also a patient advocate who served in an advisory capacity on the working group. And last, Marc, who is Deputy Director of the Oncology Center of Excellence at the FDA. So thank you all for participating. It's really a pleasure and an honor to be with all of you here today. We wanted to start with just a few minutes on each of the topics. And if we think of circulating tumor DNA as an intermediary endpoint, we think about a promising endpoint that could help speed up development and get medicines to patients sooner. But in order to qualify or to be validated, if you will, as an intermediary endpoint, we need to have a large body of evidence around its association with overall survival, which is, of course, one of the key and perhaps the most important endpoint of all in drug development. And so patient-level meta-analyses as well as trial-level meta-analyses would be required. And over the years, Friends has led circulating tumor DNA CT monitor retrospective work that's looked at patient-level results, all of which align quite nicely, indicating that decreases in ctDNA are associated with improved outcomes. So the working group reflected on that body of data and turned it to the future to prospective analyses and studies that would help address the trial level evidence base that would be required. So that said, as background, I wonder, Alex, if I could ask you to speak to your impressions of the conversations and your key takeaways from what we discussed in the working group.
Yes, of course, Nevine, and hi, everybody. Thank you very much for being here and participating in this. And it's a pleasure to be surrounded by such esteemed panelists here as well. So, I've actually worked on circulating tumor DNA or ctDNA for, I think, 12 or 13 years now. And the field is huge, right? There are so many different clinical uses for ctDNA, and some of which are already rolled out in routine use. We already use ctDNA to identify mutations that might suggest that a drug is particularly appropriate for someone's disease. Sometimes we use the detection of ctDNA in adjuvant settings to identify early recurrence of cancer. And this is because ctDNA seems to be a very good indicator of the burden of cancer in someone. And you know, at the moment, the way that we measure the burden of cancer is always indirectly, right? We use imaging to see, okay, how much disease does someone have, or we try to find a kind of simple biomarker in the blood. Well, ctDNA seems to be a very accurate indicator of how much disease, how much active disease is present in someone's body. So what we wanted to do with Friends of Cancer Research and the initiative that Nevine's referring to is beyond just sort of day-to-day use of ctDNA in the clinic. It's asking the simple question, can ctDNA measurements in clinical trials tell us about whether a drug is effective much earlier and much more accurately than we currently get that type of information? So, you know, traditionally, for example, you might test a drug in a population of individuals and look at, say, overall survival or time to recurrence on imaging. But potentially with ctDNA measurements, you could get this information really early, say within 3 weeks or 6 weeks of starting treatment. But that at the moment is just a potential. And so what we wanted to define with this white paper and this working group effort is, what do we need to do as a community to prove that ctDNA is an early endpoint that can give us all that information? And so that's actually quite a complicated thing to do. You need data across lots of different clinical trials, and that data needs to be standardized in such a way that you can compare across it. So what we really discussed in this working group was, what variables do we need to standardize? And it can be quite simple things like, we need to make sure that across trials we collect blood at the same time points so that we can compare across time points. We need to make sure that we use the same type of assays to measure how much ctDNA there is. And all this together will allow us in future to actually do a large meta-analysis and then look across all the different trials, across different cancer types and treatments, and say, yes or no, ctDNA is validated as an early endpoint in clinical trials. Does that capture everything, Nevine?
I think you did an excellent job, Alex, of going from the current clinical use to the clinical trials question to the regulatory question. So thank you for that. And Carol, I know you were at the table throughout our discussions. I wonder if we could have your personal impressions.
Sure. Thanks, Nevine. Well, this is my first time being on a working group for the Friends of Cancer Research. And I was also just learning about ctDNA. I, of course, knew something about it, but not in detail. And I have to say, it was an amazing experience for me. Friends of Cancer Research is really a unique group in terms of bringing experts from academia, from the regulatory agencies, from pharmaceutical, and including patient advocates, bringing us all together to talk about these important issues. And I was amazed, not just what I learned from the scientists in the group, but also the give and take and the amount of thought that needs to go into this. And I think Alex did a great job talking about the potential for ctDNA, the change in that to become an intermediate or an early endpoint for trials, because patients are desperate for new effective and safe drugs to treat advanced cancer, to treat all cancers. But the focus in this case was advanced cancers. And anything that we can do to bring those treatments to market sooner is something that we really need. So it was a wonderful project from that point of view. What struck me, and I think what advocates would relate to, is that as Alex was saying, there is a difference between the use of ctDNA in the clinic when we may be looking at what someone's progression of diseases or recurrence or what's happening there, as opposed to in a trial like this, a prospective trial, where we're really looking at response to treatment. So there's a little difference there, and people who are in the trial may never really know what their results were. They may, but they may not, because this is for regulatory purposes. So something that we need to keep in mind. I think the other thing is that really came to the fore for me was how there is a balance between needing to have a uniform framework for these prospective trials, in terms of when data is collected and how it's used and the assays, how they're related, et cetera. But also there needs to be some flexibility because with baseline and when people receive treatment, there can be some differences. It can be 3 weeks, 4 weeks. There may need to be a holiday for treatment for various purposes. And so to make it flexible enough to fit patient treatment schedules, but also uniform enough to fit the clinical trial is a real balancing act. And I think the framework did a good job of looking at that from a patient's perspective of how it could stay in the trial and still contribute to it. So overall, I think it has great potential to move cancer treatment forward.
Thank you for that. I think it's always good to remind ourselves that we do our work on behalf of many, many others, and your personal reflections on the process are very welcome indeed. And I think your last comments were saying we must be rigorous, but not rigid.
Right.
So let's bear that in mind as we do our work.
Perfect. Yes.
The second working group focused on a topic that I will describe just a little bit, and then I'll invite Marc to speak to it from his learned standpoint, which is we talked about the potential for circulating tumor DNA as an intermediary endpoint, but we have some today that have been qualified over the years and validated. So if we think of progression-free survival, that is a qualified intermediary endpoint. And what we've seen occur is that once the study is ongoing and we have sufficient data to draw final inference on PFS, that data is summarized, presented to the agency. And of course, there is some survival data at that point in time, but not all of the survival data. And so that survival data sometimes comes in a trial looking like it might support benefit. It might sort of be equivocal, neutral, or there might be a hint that it's harmful. The drug is harmful. And that would be contrary to what we're seeing with the PFS endpoint. And so we're left wanting to adjudicate what are the reasons for such disconnect. Are they modifiable in terms of trial design decisions that we may make? But also importantly, can we develop a unified statistical, clinical, regulatory framework that allows us to have better, more transparent, clearer interpretation of that survival data, which is interim survival data, and it's considered mainly as a safety endpoint at that point in time. So Marc, I wonder if I could invite your comments on the work that was done with the working group, but also your personal reflections.
Oh, great. Well, no, thank you very much. And I really appreciate the opportunity to be here. I just wanted to start off with a comment about ctDNA because it really kind of pairs well with the discussion about overall survival as a safety endpoint and the discussions during the working group meetings. Just in general, biomarkers such as ctDNA are very important and increasingly so for efficient and expeditious drug development, including for precision oncology efforts, that is targeting the right drug for the right patient at the right time, but also for development of novel endpoints and earlier endpoints that can be utilized. And once we have a certain level of evidence regarding that particular biomarker for use as an intermediate clinical endpoint, it really is kind of critical as an opportunity to provide drug development that's more speedy and bring these drugs to patients earlier, but certainly once demonstrating that they're safe and effective, ultimately into the hands of physicians and prescribed to patients. From the overall survival as a safety endpoint and the working group discussions and prior discussions that we've had at the agency and FDA oncology, we all do recognize that overall survival is a gold standard endpoint in oncology. It not only captures efficacy, but also safety information. But one of the misperceptions we have run into with the Oncology Center of Excellence's efforts on this initiative to capture and analyze overall survival information as a safety assessment is that we are being told by external stakeholders that we are requiring overall survival to be the primary endpoint in oncology clinical trials to support an approval in settings that previously has utilized earlier efficacy endpoints. And just I just want to clarify that that's not what we're talking about here. To really continue to use where appropriate of these earlier endpoints, such as tumor shrinkage, time to tumor progression in the advanced setting, or tumor recurrence in the earlier disease setting, because these do remain critical to demonstrate the efficacy of novel therapeutics and will continue to be important for supporting accelerated approval or traditional approval, depending on the particular clinical context and evidence. But just to note, there is this fundamental presumption that clinically meaningful reductions in tumor size or time to tumor progression will translate into longer survival or other clinical benefits, even if it's not measurable in the clinical trial. Now, that said, we have had multiple examples, which I think are important to recognize, where there is a disconnect between the treatment effects on earlier endpoints and effects on longer-term endpoints. This has really gone both ways, and we've discussed several examples during the working group meetings. As one example, novel therapeutics that harness the immune system to treat cancer. We have seen that with certain types of immunotherapeutics, there are modest effects on earlier endpoints, such as tumor shrinkage, that would not have predicted for the large improvements seen in longer-term endpoints, such as overall survival. On the contrary, unfortunately, we have seen where treatment demonstrates large effects on earlier endpoints, but at the same time, or with further follow-up, we've observed potential detriments in overall survival. So the working group discussions leading up to the annual meeting and discussion at the meeting really explored why early looks at overall survival can be challenging to interpret. For one example, an immature overall survival analysis could suggest a safety concern where there isn't really a safety concern, with the core issue relating to an inherent variability in such immature data. And the result could be that a potentially efficacious drug may be delayed or even kept off the market and ultimately not getting into the clinic for treatment of patients. But on the other hand, an immature overall survival analysis suggesting a safety concern could be the canary in the coal mine. That is an early warning signal of a potential disconnect between early endpoints and overall survival, which we have seen potentially as a result of poorly tolerated doses or serious toxicities associated with the drug, including those that could impact exposure to that investigational drug or potentially subsequent therapies. Now, we have been working with multiple stakeholders, including Friends of Cancer Research, to examine potential confounders to interpreting overall survival as a safety assessment, as well as advancing trial plans that prospectively evaluate overall survival as a safety endpoint when the primary evidence for efficacy would be from the earlier endpoints. But ultimately, we do need to ensure that the time of an approval based on tumor shrinkage, time to tumor progression, or other early clinical endpoint, we do have sufficient information and an assessment of overall survival as at least in the safety assessment to ensure the continued use of these earlier endpoints and the integrity and validity of utilizing these for supporting an approval.
Yeah, thank you for that, Marc, and very comprehensive, very precise set of comments. My takeaway from that is you're cautioning us that, yes, we want early decisions to advance drug development, but we want good decisions that aren't going to get us into trouble later on. And I think that's a very valuable tenet in the work that we all do. Thank you. And so now we've talked about clinical trials from a couple of different directions, but we haven't talked about clinical trials themselves. And the third working group really worked on this question of the burden and the complexity of trials. And they chose a particular spectrum of drug development, which is after the drug is on the market. So we've done deep, involved trials. We understand to a degree or well enough what the drug does or doesn't do in a safety profile, and it's on the market. And there are some remaining questions to be addressed. And that's where some of the more pragmatic, more modern elements can be incorporated in new post-marketing trials. And Ashita, I wonder if I could ask you first to speak on what precisely this is all about and what your experiences have been. And then Ava, we're going to go to you for your personal impressions on the conversations of the working group. Ashita.
Thank you. Thank you so much, Nevine, and thank you, Friends of Cancer Research, for the opportunity to participate in this panel alongside such terrific colleagues. I'm really passionate about the pragmatic trial work that we've been doing. By way of background, I'm a physician and a former clinical trial investigator, and I can really attest to the fact that trials over the span of my career have become increasingly more complex. And I think there's very good reason for that, but a lot of the research fields that we collect tend to be nice to have, not need to have. And there is a trade-off when you start doing that. So from the standpoint of the investigator and the patients, that increased complexity creates burden that on a practical level makes it sometimes untenable to run some of these studies at community practices and other areas where the majority of cancer patients get their care. And so by moving towards pragmatic trials, what we mean by that is if you modularize components of a trial, so the inclusion-exclusion criteria, the schedule of assessments, the endpoints that you're selecting, you have opportunities at each level, depending on the questions that you're asking, to simplify. And that simplification can have very real implications for democratizing access to research for both patients and investigators. And one of the other kind of aspects of what we were looking at is if you do, in addition to simplifying design of the trial, start considering technological enablements, you can really streamline the burden for physicians. What am I saying when I'm talking about all of that? One of the things about trials, as an example, is if you are running a research study at a location, you're hoping that the patient in front of you that may be trial eligible at the time that you're talking to them, and there may be a lot on your mind as the investigator or as the physician, that you remember that that trial is happening and that you remember to screen that patient. One of the things that we can do to kind of take that additional item off your checklist as a physician or a patient is use these technology-enabled patient finding tools. So they sit within the electronic health record. They have a subset of the inclusion-exclusion criteria, and they can send like a screen alert or a nudge at the right time to a physician and say, hey, have you thought about this patient for this trial? And that can start them down the process of a comprehensive screening and conversation and evaluation for suitability for a given study. Another thing that we can do is right now, and I think a lot of folks who touch clinical trials but haven't necessarily been involved in collecting the actual research fields and entering them into a computer don't appreciate is that a lot of times the research fields that we do enter, we have to duplicatively enter them into other systems. That duplication of effort that doesn't really add value but adds burden, we can use technology and different tools like EHR to EDC and other things to streamline that. Collectively, our experience has been, with these sorts of trials, we've been able to open community sites, which were otherwise not in play for consideration in larger trials. And we've been able to really build patient populations in trial that reflect the disease epidemiology. So typically, this tends to be more of a conversation around diversity sometimes. I think academic medical centers versus community centers, there's a difference in the proportion of minority patients there. And so by bringing trials to these other spaces, these tools, it can be very exciting and empowering. But let me stop there. I could talk entirely too long if you give me a chance. So I'll pump.
Thank you for helping to triangulate and bring us into what we mean by pragmatic elements. Ava, could I have your impressions of the discussions of the working group?
Well, thank you very much for having me here today. And it has been a real joy to be a part of the working group. Like Carol, this was also my first working group with Friends of Cancer Research. And what I really liked was the collaborative spirit and the expertise of the different stakeholders that were involved. It was just an amazing group of people with amazing insights and collaboratively trying to move forward with a perspective and both on the positives and the caveats of moving forward with pragmatic elements. What I really liked was the working group's plan to take a look at post-marketing or phase 4 settings, where safety and efficacy have already been clinically established. I believe that the incorporation of pragmatic elements would provide opportunities to significantly improve the patient centricity of the research. Because you can adjust protocol to make it as streamlined and convenient for participants as possible and incorporate additional elements to reduce patient burden. It also provides the opportunity to do implementation strategy testing since we're still in a clinical research environment. So the research team can determine how to best generate potential participant awareness and interest by developing and testing culturally competent patient and community engagement plans, which is particularly important if we're now really trying to recruit people who were under-recruited or frequently ignored in the original phase 3 trial. Together with the addition of pragmatic elements that improve the participants' experience, or at least reduce their burden, researchers should be able to sufficiently enroll the patients needed to sufficiently power key under-recruited subgroups or the subgroups of interest and address outstanding research questions, as well as find ways to improve the uptake of the improved treatment for the broader patient community. So that's what makes me really excited about this.
Great, thank you, Ava. And I think what I heard there from the third working group is let's relieve ourselves of the burden. And while we're at it, can we expand our evidence base when it comes to certain underrepresented populations and specifically do very targeted, precise questioning post-marketing with pragmatic elements. So thank you for that. So that's a quick overview of an entire day's worth of discussions at the annual meeting. We now have time for our own discussion here and the questions from participants. There's one that has come in just now, and then some came in beforehand. So I'm going to toggle between the two sets of questions. But here's a question, and Alex, I'll ask you to speak to it. And it has to do with what are the limitations of circulating tumor DNA assays? What should patient advocates know?
Yeah, it's a great question, and it's one I get asked a lot scientifically and clinically as well. And I think it reflects the fact that the technology that we use to study ctDNA is constantly evolving. So every year we have new assays come along, or the technology changes, or the thresholds change. And ultimately, what we're trying to do is find a small amount of cancer DNA in a tube of blood. And so you need, ideally, very sensitive technologies that can find that needle in a haystack and then tell you, when you look across multiple blood samples, whether that needle has gone, right? So you need kind of increasingly sensitive assays if you want to be accurate in the way that you measure ctDNA. And probably the fundamental challenge is that, well, I guess there's two. The first is that from cancer to cancer, there's different amounts of tumor DNA, of ctDNA shed. So lung cancers, for example, may shed much more circulating tumor DNA than kidney cancers. And so if you took kind of a assay that was designed for lung cancer and applied that to kidney, it might not actually work as well as you'd hoped. And the second challenge is that we don't necessarily always collect the same amount of blood. And you can imagine if you collected a lot of blood, you could get a quite accurate readout on how much cancer DNA was there. But that's not really practical to do from patients or in large clinical trials. So we typically collect a single tube. So there's always the possibility that maybe your cancer DNA isn't even in that small amount of blood. So I think it's really, it's a kind of sensitivity issue with the assays. And one thing we've really tried to do in the working group is not be prescriptive about you should use assay X or Y, but say these are the principles you should think about when you're picking an assay. These are the few variables that really matter, and some of them are things like the sensitivity of that assay. And so I think if we have that type of framework, we can really build towards comparability across trials, even when different assays are used.
Very good. Thank you for that. And now a question that actually came up a little earlier, but ties in some of the recent conversation. And the question is, how can pragmatic trials support the deeper understanding of biomarkers and their function in underrepresented populations? And there might be a few of our panelists who have a view on this. Ashita?
Sure. I think that's a really good question. And I think in the underrepresented populations, it would be important to really try and better understand where we can find patients and where they're receiving their care. I would conjecture that it likely happens a lot at community oncology sites, and that there may be differences in the patterns of how frequently they come in for their visits, and this data would be collected. So perhaps a bit of a real world pre-analysis on like how often they're coming in, and then trying to align the collection of the ctDNA endpoints. And when, Alex made a really good point about within a few weeks, you would expect to see this, response to treatment in the form of changes in the ctDNA levels. So potentially lining that up with when they would naturally be, in the window of coming in to see their provider again, and checking levels at those time points. That I think could be a really interesting way to do it. And then just thinking through stratification of patients and so forth, and really homing in on that, the dynamism of that endpoint in that subpopulation vis-a-vis, the overarching other factors that you may want to also collect. But perhaps I can pass back to Alex to comment on that and how he feels.
Yeah, I think it's interesting as well that probably the reverse question is also relevant, in the sense that we previously, sort of traditionally, we'd need people, patients, participants to come to these large academic centers if they want to participate in big trials, right? Because we need this complicated imaging and machinery that may only be available in certain areas. So I think there's a really good dovetail between ctDNA technology and pragmatic designs, because, for example, I live in a province, British Columbia, which is geographically huge, and many of our patients live days away, essentially, from our large academic centers. But if they can participate in a simple trial design where the correlative, the biomarker component, really just requires a blood tube that can be done by a traveling phlebotomist, then that really lowers the bar for their participation in clinical trials. So I think kind of this is a bidirectional question, and so actually maybe it's the underrepresented populations now better able to support biomarker development, which is a really exciting development in oncology, I think.
You said something so cool. Oh, sorry, Nevine.
Go ahead.
Alex, you said something so cool in there about having the traveling phlebotomist and enabling some decentralization elements also would like fit under that broader umbrella of pragmatism and meeting the patients where they are. And I think a blood test type assay would be perfect for something like that. There's so many great opportunities. I love the dovetail of that question.
Carol.
I was just going to agree with the example that Alex gave. I live in Vermont, which is a rural state. We're small geographically, not like Alex's area, but I know of patients who've waited a long time to be able to get into a center to be imaged. So the advantage of the blood test is just so wonderful and can really expand underrepresented groups in these studies.
Thank you for that. So I will take the chair's prerogative and add a component, which is that in studies where you've alleviated burden by introducing pragmatic elements, you should be able to make those studies large enough so that you get clarity in the subgroups and their response in terms of be it intermediary endpoints or others. So I think just an extra component Marc might have been sitting there thinking about it is we want greater representation, but we probably want a greater sample size in those subpopulations, so it's really clear what the result is.
And just to amplify, I mean, it was a fantastic example and kind of thinking, and we also have to step out of this concept of thinking about pragmatic and automatically. I think often we go to a fully pragmatic, trial. There's often, opportunities in many different trials to bring in pragmatic elements, particular aspects that can help to bring that trial, to the patient, where they live, basically. And in this issue about representation, it's so critical, and it's really about ensuring the generalizability, of the results to the U.S. population, more broadly. So critical aspect, of our kind of considerations, of the results from trials is it's applicable to the U.S. population, broadly.
Great. So I'm going to stick with the patient centricity and pragmatic trials for a second and ask for comments from the panel on patient-reported outcomes. So the patient's experience, their ability to function, how they're responding to a drug, maybe in terms of their adverse events. These are all what we call PROs. And how would those get incorporated in a pragmatic trial? Ava?
So I think that PROs are really important as an element to include as at least a secondary endpoint is to get the patient voice of the actual participants in how they felt about participating in the trial. And again, given that we're looking in this scenario at post-marketing settings, safety and efficacy have been established. So some of the PROs can be streamlined and some can be eliminated because they weren't issues that were big in the pre-formulation of the phase 3 study. And so the PROs can really be refined to get to the meat of what needs to be determined as you're doing this phase 4 testing. So I think that plus the use of digital health technologies, we can use a lot of electronic surveys and different sorts of patient-reported outcome tools, assuming that the participants want to use them, because some won't. We have to respect that as well. But we can really, again, reduce the patient burden and make their participation and their willingness to answer questions and the timeliness and the frequency of answering those questions to keep them as meaningful as possible and recognize the time and the effort that patients take to answer those questions by thanking them and telling them thanks for submission, reminding them that they need to do it, but really, really making them a key part of the research and the findings.
Thank you, Ava. Other thoughts on PROs in the context of pragmatic trials? So one thought to add is some of the pragmatic elements of trials really are enabled through technology. And so PROs are going to advance with new digital tools and kind of evolve from these big, long questionnaires with 1,000 checklist items to answer. So that will be one to watch because we'll need translation from what has come before into what the modern age will afford us for that patient voice, as Ava points out. Ashita, anything to add? You were coming off mute there.
Thank you. I think, the patient-reported outcomes are a really interesting one because I think that traditionally, and I'm probably going to date myself if I tell you how far back, but if you looked at patient-reported outcomes, you would have that clipboard that you'd be sitting in the doctor's office and you'd be filling it out, and it would be somebody else's job to kind of convert that into a format that could then be, put into a data set and analyzed. That's a lot of complexity there. I think now with digital solutions and apps, and I think even older patients are very comfortable using their phones and devices and iPads, you can sort of start inputting these in the privacy of your home asynchronously. It doesn't have to line up with a visit, and then that information can get pooled and put together for analyses. So that kind of technology-enabled streamlining is very exciting and very cool in a way to really pull in the patient voice without adding complexity. Another area where we've done a lot of really exciting work is in prostate cancer, and survival in prostate cancer has been fantastically longer and longer with the therapies that we've got. So quality of life becomes an important piece of it. And so one of the trials that we have ongoing right now, it's not a regulatory trial, it's a medical affairs trial to inform, practice patterns and so forth, is looking at, how is quality of life and specifically activity levels and sleep quality, androgen deprivation therapies, cause hot flashes in men, which is a big driver of discontinuation. We capture this information with PROs, but if we were instead able to look at sleep quality and are you waking up as much, those are meaningful endpoints for patients and ways to contextualize what happens with therapy and have those physician-patient conversations downstream in important ways. So I think all of these speak to just ways that we can be creative in trials and find answers that matter to both patients and their providers.
Thank you. Thank you. So let's switch gears and go to circulating tumor DNA. We talked a lot about in the future with new trials, we are going to work towards evaluating whether or not this is a qualified intermediary endpoint for regulatory purposes. But currently today, what role can circulating tumor DNA play in understanding indications of early treatment effectiveness? Sorry, I should say efficacy. Any thoughts on that, Alex, perhaps, or Marc?
Yeah, I think you're absolutely right that we want to build towards a time where ctDNA is a sort of confirmed intermediate endpoint, but we're not there yet. So, but most phase 2 trials and many phase 3s as well are still building in these on-treatment blood collections into their design, and they're sort of included as in some regards like exploratory endpoints. So what these trials are trying to do is understand whether the sort of early on treatment molecular response, so let's say a decrease in ctDNA or the opposite, an increase or stable ctDNA, is associated with other metrics of response. And especially in phase 2, you're really looking for signal of efficacy, and you want to kind of see multiple things that are giving you evidence that a treatment is working. Or conversely not working. It's also helpful to have quite consistent data that tell you something is not worth moving forward on. So there's sort of I would say ctDNA at the moment is being used to help qualify additional information. I did notice, Nevine, there was a question about surrogate endpoints in the Q&A as well. I think it's important to recognize that there's a technical terminology here that a surrogate endpoint is got a different level of evidence to an intermediate endpoint. I mean, it's probably something you're more qualified to speak to than I am, but they're actually quite different things, aren't they?
Yeah, and I think Marc is probably wondering if he should be hiring you to the FDA because you seem to have a handle on this. But Marc, maybe you can help clarify for the audience the terminology of surrogate, intermediary, early, in a way that would resonate for them.
Oh, absolutely. And let me just take a step back too, because to your kind of initial question about how, can ctDNA be used now in clinical trials, and just to kind of provide this spectrum, kind of a limited spectrum, but a spectrum of uses during a clinical trial, ctDNA, may be used as an eligibility criterion, for example. So patient eligibility requiring a ctDNA, biomarker finding one way or the other, could be used as a stratification factor, meaning it's a a factor that's considered when patients are going onto a clinical trial and being randomized. And the stratification is to kind of balance groups, across the treatment arms to ensure that potential prognostic or important markers are well balanced across treatment. So you kind of decrease the amount of confounding due to that. Another is, I think, came up, Alex, you brought up like phase 2 trials or proof of concept. So, a response endpoint, response biomarker. So, not an endpoint meant to support an approval, of a drug or biologic, but an endpoint to help, think about will we move forward with a particular drug development program, or how will we move it forward? Thinking about maybe treatment effect sizes and other aspects of drug development. And then we have the endpoints for regulatory, utilization for approval. Now, the one kind of endpoint we think about for an accelerated approval, which is a pathway to get drugs onto the market for patients who have serious and life-threatening diseases, for drugs that demonstrate some meaningful benefit, taking into account available therapies, an endpoint such as eventually, hopefully, ctDNA, but other types of endpoints that are typically used for, accelerated approval response rate or what have you, have a certain kind of clinical meaningfulness as an intermediary to the longer term, endpoints such as overall survival. Then what I think you brought up earlier, Nevine, in this discussion is what we call validated surrogate endpoint. So that is an endpoint that we know if you demonstrate treatment effects on that endpoint, we know that endpoint, we know that you'll see then treatment effects on that longer term endpoint that's of, clinical meaningfulness, longer endpoints such as progression-free survival in certain clinical contexts or overall survival. Now, if you think about this spectrum, the level of evidence needed for the associations and utilization of that biomarker in that clinical trial paradigm really goes up, kind of somewhat in an exponential fashion as you get to the endpoints for use as an accelerated approval endpoint, or then at the highest level of evidence as a regular approval or traditional approval endpoint. So when we're kind of in the phase where we're trying to explore, the associations and how can we potentially use this as an endpoint for accelerated approval, but don't have the evidence yet, we'll consider that as a, kind of under, as a potential earlier endpoint that needs, additional information and a lot of the aspects that I think were mentioned earlier on in terms of looking at trial level surrogacy, patient level surrogacy, and this concept of a surrogate threshold effect, which basically means, demonstrating a certain magnitude of an improvement on that earlier endpoint will then translate into a certain magnitude of benefit on that longer term endpoint.
Yeah, I think the point that you just made, Marc, which is it's exponentially more challenging to get to a properly validated qualified surrogate endpoint, is the one we don't think of often. It's not just a step change, it's a big
Very uncommon, very uncommon in oncology that we have these. And one of the things I did want to highlight for this, group is there was a recent Oncologic Drugs Advisory Committee meeting on this concept of candidate surrogate endpoints or intermediate clinical endpoints that could be potentially used for accelerated approval. There was the discussion of minimal residual disease for multiple myeloma. And there was essentially a very detailed and I think comprehensive, excellent discussion, across the committee members and the public weighed in, the FDA, that really explored, kind of what is the mechanisms for thinking about how to move an endpoint that's a biomarker that's a candidate into that intermediate clinical endpoint realm for potential use or earlier clinical endpoint for use as accelerated approval. I can provide the link in the chat in case those are interested to the meeting materials and can look at that online. But I think it was an excellent discussion. And for those who are, very interested in this, concept, that really goes through very nicely. What are those evidentiary kind of, requirements and what does it actually look like? Because that was the bulk of the discussion for MRD and multiple myeloma.
It's years and years. Thank you for that. There's a question that's come up, and it's back to the pragmatic trials, and it's a very specific question, but one that helps bring the concepts to life. And it's a question of if, and people talk about this a lot, one of the pragmatic elements of the trial could be using existing data in an electronic healthcare record, so data from any patient's encounter with their medical team, wherever they may be, in order to invite them to be recruited to a trial. And I wonder if one of our panelists could walk us kind of step by step. What does that look like? And maybe even how does it differ from today's or maybe yesterday's clinical trials? Ashita?
I'm happy to take that one. Thank you. So it's a really good question. I think when people think about the electronic health record, we think about just it in its kind of current state where, you go see your doctor, they'll enter the information into the EHR. It may not necessarily cover the entirety of all of the research fields that you would want to collect. So missing this is a challenge. And then on top of that, when you come in to see your doctor, naturally may not, in most cases, will not really line up with the very prescriptive schedule of assessments in a clinical trial, which for time-based endpoints like OS and MRD and ctDNA and what have you, that can really matter. So essentially what we do for the EHR to EDC component is there's a couple of different flavors of this. It really depends on what is the electronic health record being used at a given site. So some sites use Epic, some sites use Cerner, some sites use like OncoEMR, and there's like a plethora of different EHRs. So having that understanding of what is being natively used is really kind of like an initial filter in your site selection. And not every site in your trial may leverage this technology, or you may need to have several different strategies if there are several different EHRs in your site footprint. But if you want to simplify for a second, let's say that you have the same EHR. There are now, you come to a different node in your decision tree. You can use whatever subset of your fields are native to the EHR and overlap with what you're collecting for your research study, and have those fields only port in and have some provisions in place. And these are all, again, for privacy purposes, patients would be consented into the trial. All of their data would be protected. It would be transferred, and analyzed in a very secure environment so that these data breaches aren't really meant to be a concern at all. This is all done with, full visibility for investigators and for the patients themselves. And then subsequent from that, okay, so that data that's coming in, what we would do after that is we would essentially, have site support staff to ensure completeness. So that's kind of one way with the native fields. The other thing that you can do, and this is really kind of future forward, very innovative. We've done it, but I think we're probably among the first studies or companies to try and do this, is for a subset, for a particular EHR, we're able to work with the EHR vendor and essentially create new research fields and integrate them into the EHR that mirror one-to-one what we would like to see collected in our trial. And all this means is that that information that maybe was captured in an unstructured free-form text, now we've created structured fields, and we've got site support staff to ensure that the fields are being populated, and the physician can just right there and then in front of them at the point of care, enter that data, and it'll get piped seamlessly into the analytic set for analysis. Now that sounds complicated, it's even more complicated to make it happen on the back end, but it's the direction of travel. And I think what makes it exciting. So when people say EHR to EDC, there's a lot of different views, flavors, and practical concerns that go into it.
Thank you.
Eva, do you want to add anything, as particularly around privacy?
Well, I was thinking that when you actually select the sites, like for a phase 4 trial, let's say that you need to improve the representation of African Americans with a certain disease in the trial. When you select the sites, I imagine that then you're looking at the big data of the number of patients that have the disease in different sites to, it could be a quite different group of sites that you're using as primary sites for phase 4 research than you did in phase 3 research.
A little bit. So I think regardless of the phase of the research, there's always pre-work that is done using real-world data to kind of help inform the site strategy. And it is based on, these trial eligible patients, and certain sites, for whatever reason and reputational like elements of the providers that are there, may tend to see more of a certain type of patient. Like you may have the world's expert in, this particular mutational factor in lung cancer at this subset of sites that are going to draw in more patients, and it would make sense to activate those sites. So that sort of site strategy happens upstream, but in a lot of trials.
So I'm just looking at the time, and I think we're going to go ahead and wrap up. What we've heard today is the promise of circulating tumor DNA. Certainly sponsors can, and other researchers can use this promising endpoint for stratification, for patient selection, for decisions early in drug development, like dose levels and whether or not to proceed. But there is a exponentially bigger piece of work coming to qualify it or not. We don't know what the data will tell us as a surrogate endpoint. We heard about the challenge of having limited survival data when surrogate endpoint is used as the basis of approval, and how the agency and others are developing frameworks for that to be done in a comprehensive manner. And we've talked quite a bit actually about pragmatic elements in post-marketing trials that help bring the trials closer to the patient and also alleviate burden at the research sites. One thing we didn't say explicitly, but I think you're all aware as members of the audience, you are our surrogates when it comes to speaking with patients and physicians. And so anything else we can do to help you reflect on these complex topics that we've talked about briefly today, don't hesitate to reach out. And having said that, I'll thank the attendees and the panelists for a great hour and hand it back to Hillary for closeout. Hillary.
Thanks, Nevine. And thank you to all of our panelists for participating in our event. Also, many thanks to our audience for joining us today. If you're interested in learning more about the drug development process, please consider taking our course at progressforpatients.org. We'll be sending around a survey about this event to help us improve our advocate-focused events in the future. Please fill it out or email us at info@focr.org with any comments. We hope you have a great rest of your day.